Welcome to Module 2. This is a companion video if you couldn't make the meeting, or if you need some help with your own gene, if you're doing this on your own or checking your work on your own. Please make sure that you've done Module 1 first, because you really can't do Module 2 without Module 1. So go into our files on our team site and check that out. I am in GENI-ACT, and I am at the point where my screen looks a little different than yours, but I'm looking for that demo gene that we're going to go through in this video so that you are prepared to do your regular gene. You should split screen your screen or pull up this video on your phone so that you can devote your screen to what you need to do in your notebook and pause as needed with the video. So I'm going to open my lab notebook, which is under the lab notebook, and I want KSED 00010, the demo gene that we started with last time we met. As we add more information to the notebook, it'll take a little longer to open, especially if you have a slower internet connection, so just be patient. So last week, we really did one thing. We did the basic information about this sample gene. You did the basic information about your Clostridium gene, which again is unique to you. You are the first human most likely to be looking at that gene and researching it. So we're going to go back into that, clicking the plus, and you can see the stuff that we added in last week. It took you guys about 5 or 10 minutes. And I scrolled down to the protein sequence because really for the rest of this study, we're going to come back to this again and again. If you look at the letters in DNA, it's A, T, C, and G. But if you remember, there are multiple ways to get to a specific amino acid. In other words, one triplet up here might code for one amino acid, but a different triplet could code for the same amino acid. So there's a redundancy up here that we're going to avoid by just using the amino acid sequence. And just a point before we go ahead with this, each letter here represents an amino acid. There is a code for it that's in your training manual. You don't need to memorize it, okay? But for example, M is methionine, S is serine, and each one has its own letter code. So each one of these is an amino acid, not every 3. I am going to do some things to prepare. First of all, I'm going to get another tab open, and I'm going to go to, in this case, it's my website, okay? So Benedict's website, and right up here it says Bioinformatics UB Research, and you're going to click it. I would strongly recommend you favorite this so that you don't have to repeat this for every tool that we use. You'll scroll to the bottom and you can see there's a link to BLAST. Module 2 BLAST search compares your amino acid sequence to amino acid sequences from other sequenced genomes. So go ahead and click it, and I have it programmed to open in another tab. But before you do anything, copy this. I just Control C, and I made another tab where I'm going to go to the same place. There's a reason for that in a second. Before you go any further, I would strongly recommend that you sign into the browser, so like sign into Chrome or into Firefox, and favorite this site. If you don't want to do that, favorite the site in an email to yourself so that you can favorite it at home on whatever computer you're using. So for both tabs, now that they're open, I'm going to click Protein Blast. So I'm going to the other tab and I'm going to hit Protein Blast. And it opens a window that looks like this on both. I'm on the same website. So now I'm going to go back to my GENI-ACT notebook, and I'm going to copy with the header my amino acid sequence. Control C. I'm going to start the first one off because it takes longer. I'm just Control V to paste it. It looks a little funny. I'm going to try it first and see what happens. This first one that takes longer is going to be the non-redundant protein sequences NR database. When I click BLAST at the bottom, it's going to take my amino acid sequence that I pasted in up here, and it's going to run it against all other known amino acid sequences from proteins that have been sequenced. That sounds great, except that most of the proteins in the NR database have not been confirmed. They're like the work that you're doing on your gene. They probably maybe could have a certain function, but we don't know yet. So this is one test we're going to run to see if other proteins are out there that are similar to this KSED 00010. So I'm going to go ahead and get that blasting. I'm going to go back to the other side, to this other tab, and I'm going to paste in the same amino acid sequence, my gene, this 00010, and this time I'm going to pick a different database. And I'm going to pick Swissprot, which is a database that is much smaller, but it represents proteins that have only been tested and confirmed in a lab. So we know a lot more about them. So now I'm going to be comparing my protein, or in this case, KZ00010, against known proteins that are out there that have been studied in a lab. I'm going to click blast here too. This one's a smaller database. It's going to go faster. So you very well may find that one of these beats the other. And in this case, you can see my Swiss prot is already up, and it looks kind of like this. Okay, so we'll talk about that in a second. I'm going to start with the Swiss prot database because the results are going to be more high quality because the genes have been studied in the lab. So it's got some stuff up on the top that we'll talk about later. Then it's got alignment scores. So it says distribution of the top 106 blast hits on 100 subject sequences. Okay, so if you look and hover over each bar, it tells you chromosomal replication initiator protein. Same thing. And you go down and you can see these ones that have almost 100% match or alignment are all kind of saying the same thing. Chromosomal replication initiator protein. Down here where things start to fall off, where there's not so much alignment, 40 to 50 in the blue here, this is still a chromosomal replication initiator protein, but it's from a different organism, one that's not so related. So in terms of quality, there's proteins out there in this Swiss prot database that indicate that my protein is similar to them. So that tells me something immediately about what my protein might be functioning as or what I could name it. This is the NR database. You can see it doesn't look the same, but there's still, for KZ00010, there's still full length red bars, which means I've got some really good alignment going on. So I've got some data. So what I'm going to look for, I'm starting at Swiss prot, and I'm going to go down and I'm going to look for a few things. I'm going to look at the score, the query cover, and the e-value, and as well the identity. This is how you interpret it. It is in your training manual. In general, I'm looking for e-values that are 1 × 10 to the -3 or smaller. And what the e-value is the chance that the similarity in the sequence is just random chance. And in this case, it's 1 × 10 to the -179. That's the chance that it's just a randomly, it just matches and they're not related at all. So in other words, it's probably not random. The two are a good match. So you can see the names of all of these are all the same. Okay, so I probably know what I have from KZ00010. There's no variety here. And if I go to NR, let's see if it says the same name. Chromosomal replication initiator protein. So between the two databases, I have a lot of information that says, yeah, it's probably a chromosomal replication initiator DNA-A. So if I look at the e-values here in NR, I have 0. So there's a 0% chance that this is a match just based on chance. Now what you have to be careful of in NR is this first result, because look at what it is. It is the gene I just put in. So of course, it's going to match the gene that I put in. And it's saying that it's 100% the same identity. And if I look down here, the other 100% is from my bacteria, Chitococcus sedentarius. I need to kind of ignore those. So I have significant results from both. The score is going to be telling you how well they match. So a higher number is better. Okay, and that's really all you need to know. The query cover is how much percentage-wise that the two proteins match. So in this case, the query cover, 99% of the two proteins kind of match each other up, and there's a 0% chance that that happened by accident. And look, I'm in a totally different organism, Serinococcus profundi. That's not even my Chitococcus sedentarius. So I've got some data I've got to provide for my notebook. So I'm going to go back to my Swiss prot tab, and I'm going to double check. It does list it right here. I'm going to go to Swiss prot, then I'm going to go down, and if I look at my first example here, or my first result, I make sure that it's not the same organism, so it's not 100%. My e-value is really low, and it does cover a lot of the protein, so this is a good thing. And this would be a good result to click. So I'm going to click it, open, and I'm going to start getting some things for my notebook. So this is the specific alignment between my Chitococcus sedentarius 00010 and my sample gene. It's the alignment between Chitococcus 00010 and this organism's gene. What this is showing you, if you look at the line between what your query was in the subject. My protein, Chytococcus sedentarius 00010, is the query. So you can see it's just filled in. That's what I copied and pasted, query, and it just goes line by line. And the subject that I just clicked is going to be listed here. What you see in between, the pluses and the letters and sometimes a gap where there's just nothing, is what the match was like. So if there's an exact match, you will see that letter. Okay, so like right here, EEE. It's a perfect match right there. And you can see that happens quite a bit through here. There's a whole region here, A's, H's, A's, and F's, and R's, and N's, and S's, that match perfectly between the query and the subject. Okay? But sometimes there's not a perfect match. So if you see a plus sign, what that means is that there's an amino acid there that has some similar properties to the A amino acid, but it actually was S. So it's not the same, but it's similar. So the plus is okay. It's still possible that it's related. No letter means there's a complete mismatch. So you can see right here there's an A, but in the subject it was a T. So that's not going to be alanine versus threonine. That's not going to be a match. Okay. BLAST will also, to massage the match, it'll introduce some gaps. So if you see minuses, that means it inserted a space to make something later in the protein line up a little bit better because it thought there was a little bit of a match there. Okay, and that might come from a gene being inserted or deleted sometime in that bacteria's history, which is kind of cool. You'll want to click, the next thing is a GenBank. Up here, you'll see some hyperlinks, and if you click this one, really, or this one, and I just did, it opens a gene bank record for this protein. And remember, what we got from SwissPro was the result that I just clicked, the first best match, KSED 00010. Okay, so it's the best, highest probability match. And we have a name right here. We also have some stuff down below, like what organism this comes from. It does also talk about a little bit what it's thought to do. And since this is SwissPro, it's been confirmed or used in a lab. So this is not somebody's random guess. This is actually something actually accurate. And it even comments on what it does. It plays an important role in the initiation and regulation of chromosomes replicating. Okay, and it has a whole bunch of other details. So it's really cool how deep they can go once they get this gene into a lab and study it. So anyway, I'm going to head back to my notebook, and you are going to be in, this time, the sequence-based similarity data. So make sure that's clicked open. The link to BLAST is always here if you need it. Now the gene product name, the top hit, is what we're going to be doing. So remember, you click the note icon and it makes it editable. Now the gene product name, if you check the key that I've provided to KSED 0010, you'll note that they dealt with this in a particular way. And they have a note that says, basically, in this first hit, or gene product name top hit, they actually include two. And because SwissPro is more reliable, they're going to include information about the top two hits. Later, down the page where it says hit number 2, alignment of the second hit in query sequence, they're going to take that stuff from the NR database, which is more unconfirmed, okay, and less certain. So we're going to follow the same pattern. So the gene product name, top hit, I'm just going to make a note because this is a notebook that other people would want to follow. And so I'll make that note right now. I need the name of the hit, the gene product name, and that would be this, okay? When in doubt, take the whole thing. Record name, full is that. The actual name is chromosomal replication initiator protein DEA. So I copy it, I go to my book, I make sure I'm going to hit 1, and I paste. You don't have to worry too much about fonts unless it bugs you. I just went backwards in it and made it the regular font. Okay? It's magic. I don't know how it happened. So I'm also going to record the second hit here. And I go back. So I'm going to come back and I'm on this page again. And all I need to do is come down to the second hit, which is right below it. And I'm going to click open this GenBank information page. It opens a new tab automatically for me. And this one has the same name. So I'm going to copy and I'll paste in a second, but I'm going to make sure it's not from the same organism. And if I look, the organism is not the same as my other one. So it's not like this is messing up and telling me 2 of the exact same gene from the exact same organism. This is different. It's a gene that is also in that other organism. So we're looking at some kind of conservation amongst different species. So we've got hit 1 and hit 2 for the gene product name from Swiss Pro. And I've got those two tabs still open. The next thing I save, have to save, okay? I go down, organism. Well, that's on those two pages. So I want to make sure that I keep these straight. Hit number 1 and hit number 2. Okay, so just to be accurate, before hit number 1, I got to keep track of my tabs, right? I just happen to remember it was from GENI-ACT radiotolerance. This one is not. So this is my hit 2. So I'm going to paste that for hit 2. For hit 1, you'll notice I don't have a tab up there anymore. Somehow I used the tab for something else. So what I'm going to do, it's very easy to get back there. I'm going to go back to my first hit and hit the link again. Okay, so it substituted it. And I'm back to GENI-ACT radiotolerance. And I'm just going to take the whole thing, paste it into my GENI-ACT notebook, make it pretty. This is online. Hit save. Okay, so I've got my 2 top hits for a protein that is very similar to mine, like highly similar, probably well conserved. Now it says the alignment length, and I got to go back to Swiss Pro, click the notebook to edit it. Remember, you can always scroll to the top, and it'll tell you here if it's Swiss Pro versus NR, and we're still in Swiss Pro. So I'm going to scroll down. It has a bunch of information along the top, some more statistics. Now the alignment length is a bizarre thing, okay? In general, remember that your protein is the query. The subject we're comparing it to is the thing that the Swiss Pro database and BLAST said, hey, there's similarity here, we got to check out. So you'll notice that the similarity starts, or the alignment starts at amino acid number 11. It ends at amino acid number 503. So as far as your alignment, there's a funky rule. The alignment length is going to be equal to the number of the last residue from the query, which in this case, 503. I just get the number at the end, minus the number of the first residue, which is number 11. It's the 11th amino acid in my sequence, plus 1. So mine will be 503 minus 11 plus 1. So 503 minus 11. So 503 minus 11 equals, but you got to do the plus 1, okay? If you need to see that special math formula for yourself, that is on page 39 in the training manual, okay? But what that tells us is what the whole alignment length actually is. It's not 500 amino acids long that this computer was doing the match with. It's 503 minus 11 plus 1. So it's actually 493. And that is hit number 1. So I'm going to my GENI-ACT notebook, and I'm going to keep them straight again. Hit number 1, 493. Hit number 2, I'm going to get to the other record. So this is my first one. I go to the second one, and I'm going to do the same thing. 504 minus the first number, which is 1. So it would be 503, but then I'm going to add 1. So it's 504. So I go to GENI-ACT. Hit 2 is 504 amino acids long. Save. Easy, right? Now the scores, we're back to familiar territory. Okay, we do have 2 hits. We're going to do hit number 1. Go back, make sure we're in Swiss. The score, I'm going to steal it. 517 bits, 1331. Hit number 2. I'm going to go back, and I'm going to scroll to hit number 2. I'm going to take it. It's got a little bit lower score. Higher score is better match. Okay, so this one's a little bit lower score, but it's still quite good. And I'm going to save it. E-value, same thing. I'm going to take hit 1, scroll back up for it. My E-value is the expect value. Remember, this is the one that determines the chances that it's just random chance that these two alignments are so stinking close. And the chance of that is almost 0%, okay? Because it's 1 times 10 to the minus 179. That's a really small chance that those are related just randomly. So I go back for hit number 2, copy and paste that expected value, which is a little bit bigger, but we're still talking ridiculously close to 0 in terms of the chances that that's a random matchup. It's probably non-random. Alignment of the top hit and the query sequence. Now, this might be a first-time thing for you. This is easy, but it is definitely a 21st century skill. So what I'm going to do, with hit number 1, I don't want to just have those details. I want this picture that I can open up my notebook and get back to anytime. So if you go down to the bottom and you press snip or type in snip, you're going to find the snipping tool. This is Windows 10, but the Windows programs anytime recently have this. You just have to find it. It might be in your tools or system tools. And you're going to click new, and you're going to do a rectangular snip, and it'll gray out the screen and give you a cursor. Okay, and all you have to do is basically drag the window so that you get this data. Let go, and it snips it for you, kind of as a screenshot. Okay, once you do that, though, you're not done. You do not want to copy and paste that. Okay, it is a picture, and we want to save it as a picture. So save as format. PNG is very easy for GENI-ACT to deal with. So I would recommend you use it. Okay, I would also recommend you don't just name this capture or blast, but name it something good, like KSED 00010 blast, match 1 blast. That way, if you come back and you're trying to find the thing again, something crashed, or you forget to press save and you find out later that it's blank, you just have to go into a folder that you have in your computer or a thumb drive or your OneDrive or your Google Drive, and you find it and you just bring it back. Somebody you could see on my computer labeled there's thing and thingy. That isn't going to be very easy to find later, so I would not do that. Okay, this is really specific. KSED 00010 match 1 blast. Okay, and we're gonna save it. I'm just putting it on my desktop for now because I'm gonna get rid of it. We go back to the notebook, and this time instead of trying to type something in, we're gonna browse to upload an image. And I'm gonna find that one, open it, and it's saying when it lists it here that it's ready. It found it, but I have to press upload. And look, there it is. So there's my hit number 1. For hit 2, I don't mess myself up, scroll to the second one, and I'm going to do the same thing. Close it off. I'm going to minimize the snipping tool this time because I'm going to end up using it again. Now I need to browse for this new one. Okay, so you could see what I just messed up. I did forget this. This is interesting. This will help you watching me screw this up. Whatever you put in, whatever you upload second will be on top. So I'm going to, just for neatness, I'm not going to bother with the headings anyway. But because I want this match 2 to be on the bottom, I'm going to upload it first, and there it is. Now I'm going to browse and get my other one so that match 1 is on top, because that's what's happening in all my other windows. And I do want this to be easy for me to read as well as other people. So I'm just going to confirm. I remember that my hit 1 was to the negative 179. So I'm just going to, there it is right on top. And if I scroll down, I can resize this window, which I am because it's going to drive me crazy. There's my other one. Let's make sure I don't have any other junk. Nothing. Good. Save. Keep coming. Now I'm into the second hit. And as I said, what they did with the key here is for the second hit, which is technically the third best, they went to the NR database for this one. So if you take a peek here at my NR results, because I haven't used it all so far, I scroll down, it really does look the same. It's got that same name, chromosomal replication initiation protein DNA A. And there's my species there first. I kind of need to ignore that because I know that this is probably my gene. There's a couple 100% identity matches. That's telling me that this is my protein that it's matched up to in the database. So I don't want to compare my protein to see if it's similar. It's 100% similar. Even this one is from the same species, so I'm going to try to ignore that. So this one right here, though, is not the same. So that's kind of the first species that's not along with my genus, Chytococcus. So that's the one I'm going to try to zoom in on. If I click it, takes me right down there. Okay, and I double check. I'm not even going to say it. Okay, a different kind of bacteria completely. Now I'm doing the same thing. So I'm figuring out, I hit this link, and it brings up the GenBank record. And it gives me, lo and behold, the same name. So back in my notebook, I'm going to do hit number 2, the second most. And I'm making sure I'm in NR. I am. If I go back up here, the second one that seems to be there is kind of right below it. It's still got an e-value of 0, and it still covers most of the alignment. So this is still pretty good. So I'm going to click that next. The organism, same deal. We're going to keep them straight though. I'm going to click it again just for kicks. And here is my species, my bacterial species. GENI-ACT, hit number 1. So here's my second hit. It's right under the other one, so I'm going to click it. And I'm going to do the same thing. Hit the GenBank record, go to that tab. Here's the name. So I'm going to take it, put this in the book. And fix this up here. I don't know if you saw what I just did, but I missed the proper one. I didn't go right underneath the other one. So I'm just fixing it. It's the same genus. It's probably not a big deal. We're going to make sure that it matches perfectly. So what I did basically, I mean, you are going to run into the same stuff. Look carefully. I was not being careful. So this was my first pick that was not Chirococcus. I don't want to compare it to myself or to my protein that I'm looking at and studying. 00010 is from Chirococcus species. So I want to ignore those. So my first one was this, but then I saw this next time, so I picked the wrong one. These two would be the highest up on the list, so these would be better. And they just have higher numbers all around. So that's what that looks like and why I did that. And now I'm on to the next thing. I want to make sure I save the organism I fixed, but I'm going to save them. Now the alignment length is the same deal as before. Hit 1, hit 2, and now I'm going back. And this is that weird math problem. So if I hit this as my shortcut, this was my first one. The alignment length is 504 minus 2 plus 1. So 504 minus 2 is 502 plus 1 is 503. So my first hit is 503 amino acids long. My second hit should be right below it. Yep, Serenococcus profundi is 504 minus 1, 503, plus 1, 504. So that one's an easy one again. 504 amino acids long, save it. The scores, same thing. Hit 1, hit 2. I'm on hit 2, so I'm going to record the score for hit 2. I'm going to record the score for hit 1. And this is the right one. Save. P-values, same deal. This is my hit 1, 0% chance that the match is completely up to chance. For hit 2, it is similarly 0. So there's something going on here. They're similar for this sample gene. Save it. And now I'm going to have to get, finally, my pictures. So I'm going to go back to my snippy tool, and I'm going to go back to NR, and I'm going to make sure I have my second one first, right? Because it's going to put whatever I put in there first, it's going to put towards the bottom, and it's going to keep shoving it down as I add other pictures. So I'm going to get the hit number 2 snip first. See, I'm learning. So, oh my gosh, new. And once again, I strongly recommend you save this as an actual name. I could have put Swiss Pro Blast, and this other one would have been even a better title. Okay, and actually while I'm here, if you need to do that, you can rename it. Because you use these throughout the rest, whatever detail you can give is better than, what is that file that I have? Okay, so we've got case at 00010 and our match 2 blast. Save. Go back. Browse for it. Upload, and then we're going to just do the same thing. I didn't snip it yet though, so I've got to go back to it. New one. I didn't scroll first, so don't put the same thing in twice. And I have to go up to my first hit, this one, the Pekingans. Get the snip back, so I closed it. New snip, rectangular. Save this also. Try to follow the same pattern. Case said 00010. And on match 1 blast. Save that. Browse for match 1 this time. And press upload. And then we gotta make sure that it's here. So here it is. It's right on top, and it kicked the other one, which was my hit 2, downwards. So I'll make sure that it's there. There it is. Save it. Even if you upload, you need to press save. And that brings us to the end of the blast sequence. You're going to run these a couple times. So we took our time to make sure we did this example right. There is an additional sheet if you have trouble and you run your Clostridium gene and find that all your results are Clostridium. So check out that for that thing in the files if that happens to you. And there's basically a simple checkbox to exclude Clostridium so you don't have to sort through a lot of stuff. Clostridium is hot in medicine right now in terms of research. So a lot of people are messing with it in the lab, and you'll find a lot of stuff in NR and SwissProt about it. So it's kind of cool. We're right at the cutting edge here of science. So give it a shot with your Clostridium gene. Please come see me or send me an email if you have trouble. But in general, the key to this Chytococcus sedentarius 00010 is also in our files. And I would recommend that you kind of have that open or print it off for yourself nearby so you can see that you're following the right pattern and providing the right info. Your training manual also kind of goes into a lot more detail than what you just went through with me. So if you are lost in the sauce, use your resources, including me, and we'll help you out. All right, good luck.